Welcome to the Atlantico podcast, where we talk about the science behind the Atlantico project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of Atlantico and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. Today we have another episode in our Atlantic Ocean series focusing on one of the specific topics of the mission microbiomes of the Tara Ocean Foundation, one of Atlantico's flagship expeditions. Our guest today is Erica Becker, who was on board of the biodiscovery leg in the Trinidad Seamounts along the coast of Brazil. In our conversation, we'll hear from her experience on board Tara and get an idea of what scientific questions the team looked into getting an answer during the leg. Welcome to the podcast, Erica. I'm happy to have this conversation with you today.
Hi, Eloise. My pleasure. It's so nice to be here and talk about this amazing leg, which was one of the core legs that we have Atlantico's mission in Brazil. So let's talk about science, let's talk about South Atlantic.
Yeah, brilliant. We like to start the podcast by learning about our guests. So I would like to know a little bit more about you. Can you tell us about yourself, where your connection with the ocean comes from, and what your journey has been to get where you are today and to do what you do today?
Yeah, sure. So I am a marine biologist and a PhD research at the Federal University of Santa Catarina in the graduate program in ecology. Like any other kids, my passion in the ocean is more about the curiosity to understand where we are in nature, how things work. So I'm curious about the infinity of life present in one drop of seawater. I'm seek to understand how very tiny planktonic organisms are related to the function of the ecosystem. So in this context, for the past 10 years, I've been working and pursuing a career specializing in marine zooplankton. So yeah, ask me about copepods. Trust me, they are amazing. They have a very high taxonomic diversity, different ways of feeding, of behaving in the ocean, and they are one of the most abundant animals that we have in the earth, so they are very interesting. Actually, I just finished my PhD research and for this I've been working on patterns and drivers of functional diversity and the size structure of marine copepods, and investigating how they are related to the function of the South Atlantic Ocean ecosystem. So my PhD is very related to the project of Atlantico. I've been applying for the past years a functional and taxonomic diversity index, modeling the size distribution of copepods from imaging data in a very complementary way, merging size and species-based approaches. So I'm also have a son, Miguel, and I love the whole universe of around the empowerment of woman scientists and mother. And I also like to be involved in biodiversity conservation projects, combining art, science, and environmental education.
Nice, that sounds very interesting and indeed very connected to what is being done in Atlantico. And if I can ask just another question, where did your love for the ocean start? So what was the story there?
Yeah, I think going on vacations, so those very small time frames that we have during the year when you have a break with your family and you are in a very comfort zone to actually enjoy. I think I have the best memories of my life just exploring and looking the ocean, the animals, the sand. All of this sounds so nostalgic for me. And when I realized where I was in the world, what I want to pursue in life, I start to make questions like, can I do this as a living? So I started to look after it, and actually this is a passion coming true, a dreaming coming true, I guess. And for example, I see for the past 10 years that I've been working on this, that you can transform science in other ways of communicating and doing it, and not stay only inside the academia. So this is also something that we are doing right now, and also I'm starting to enjoy.
Brilliant.
So I never want to do something in life that was very boring, or I have a fixed routine, and I think in science you can walk in different areas and you can explore all the best you can do for the world.
Excellent. So let's talk about your experience during one of the topic studies of mission microbiomes. You were on board of Tara from Salvador de Bahia to Rio de Janeiro to study the biodiversity in the, I'm going to let you probably correct me on the way to say that, but in the Vitoria-Trinidade Seamounts chain. So let's start by discussing the specificity of the region and the scientific questions you were addressing during that leg. So we want to understand why studying those things are important and how it will contribute to increasing our knowledge of the Atlantic Ocean and its evolution. So can you tell us all about it?
Yes, so after leaving Salvador, we navigated for about 3 weeks. So it was intense days of navigation between the start until the end. And drifting in the area and actually doing the work, it was 11, 12 days. Drifting in the seamounts, which is composed by 11 seamounts. So a seamount, different from an island, doesn't reach out to the surface of the water, right? But however, they can behave like islands in the ocean. So by this I mean it might create unique oceanographic features as obstacles in the prevalent current along the water column. In this particular area, Vitoria-Trinidade chain in the South Atlantic, I think what is more impressive is that you can find some tips of the seamounts very close to the surface of the water, like 20, 30 meters. But it is very steep, so just going away just a few meters means you're reaching 2,000, 3,000 meters of deep. Then that's why it's a very unique and amazing place where Atlantic wants to do the same thing. Because it presents a remarkable opportunity for studying the ecology of marine life. It's an oceanographic signature that creates the water, together with the fact that it's very isolated from other places, other islands. They enrich the water column with nutrients and allows the ecosystem to develop, including unique species that we call endemic in the area, which means that only occur in this area. So it basically describes a hotspot for biodiversity. That's why most of the science that are very lucky to be there, it's not only for foreign scientists, but also for Brazilian. We are very proud to have this area as a Brazilian territory. But it's far from the coast and particularly protected from human activity. So it's very special. All the scientists, they want therefore to understand the impact of seamounts on biodiversity. Usually the most of the studies that we have in these areas is targeting to understand the biodiversity that is there. Considering this, this was the biodiscovery leg from the mission, which is part of one of the Atlanteco's case studies on molecular bioprospecting. And we have a few milestones to reach on this leg. So increase plankton biodiversity discovery. So we replicated a lot of sampling, higher vertical resolution, sampling more frequent, before the seamount, after the seamount, in different current directions. And then to see the potential of discovery of new bioproducts and chemicals for possible industrial applications, and that has a high socioeconomic value For example, new ways of producing pharmaceuticals or natural enzymes which could be able to digest complex molecules such as plastic or any other pollutants that is present in the water. And also I think the macro scale ambition is to measure the impact of climate change, but especially the impact of plastic that is so important nowadays to have an overview what's going on in the ocean.
Yeah, I mean, a very ambitious leg with a lot of objectives, and it sounds like a very rich region with a lot of life. So when we talk about biodiversity, we talk about many different forms of life, different animals, different plants, I guess, and from all different sizes, very big to the invisible. And biodiscovery, it's discovering those new forms of life. And bioprospecting, as you said, is how to utilize those forms of life for, in some cases, pharmaceutical applications or things like that. So can you tell us how this was studied on board of Tara? What did you have to do? And then what will happen next with those samples and the data?
Sure. So first of all, we have this very effort to replicate, to done a lot of sampling, thinking about what is the perfect strategy that we could have in the area, considering the oceanography, the physical, the chemical, and the biological background. The second one was to do a very fine scale characterization of the observed deep chlorophyll maximum, or DCM, which is the region just below of the surface where we can have this signal of the maximum chlorophyll produced by the phytoplankton. So we also sampling stratified waters at both long stations, which included several layers in the water column, as well as overnight sampling. So it was a very nice work in the sense that we could actually be in there and exploring any kind of methods or strategic that we could imagine that was possible in this issue of biodiscovery. So we always have this in mind in our discussions after lunch or in the morning or just before work. So it's an amazing environment to be planning this kind of stuff, because you have everything prepared in the project, but the project is just the theory of what you're going to do. And then we are in there in the ocean, so you have your life moving, you have everything happening, you're preparing a lot of things in the day before to sampling. Then this is something else. So it's amazing to how you can see how the sea is now today, so what we can do, how many hours in the day. It's so many questions that you have to keep in mind. It's so expensive to be in the ocean, it's so far away, you are far from your family, there's so many variables to be there. So we need to be very focused in what you are doing and what is the initial proposal. So
a lot of adaptation. So
yes,
you're there, you have the theory, or so the planning that is done from the land, the people in front of the computers.
You have last minute stuff, you have people that are being seasick, so you have to always take care of each other. But coming back here, by this overnight sampling, we expect to find more abundance and more species as a final ambition for this sampling. So to be as exhaustive as possible in our study, in our goals, we set up a night station from 7 p.m. until 5 in the morning. And we sample the same 7 different layers that we have sampled during the day, so we can compare. To observe the migration of the plankton, which is the most important feature that they have, and they do it in a daily basis. So this refers the way zooplankton moves upward and downward every single day. So during night, they rise up towards the surface to feed on smaller particles, smaller phytoplankton. This great migration steers the water column and modifies chemically, physically, and creates an exchange of matter between different layers of the water column. So during the sample, I was in charge of filtration systems. So we set up a line of filtration with different size of filters to do microplastics, EDNA, particulate matter, mercury, and I also collect water to do biogeochemistry analysis. What is interesting to mention about the EDNA collection is...
So before you go into further detail, can you just explain a little bit what EDNA is?
Sure. So eDNA, it's an environmental DNA, so it's quite recent the way we study this, and it's in a community level. So we target with this analysis any imprint of DNA that can be tracked in the area. So we want to know what is the microbiome, but also we want to know what is the biological interactions we might have in the area. So eDNA, it's important because we collect in day stations and also during the way while we are navigating between stations. So this is data that we want to compare with optical properties of the water that have been collecting real time in the underway lab. So it's a very integrative, our view, our snapshot of the ocean that we're going to have is not only about the microbiome, but also the other scales of biology and optical properties, physical, chemical. So it's huge. It's massive.
It sounds huge.
The data that we are going to produce, and also our understanding of the complexity of planktonic ecosystem. And then finally we properly preserved and shipped these samples to the partner laboratories on land to conduct the genetic, microscopic and other analysis.
So a lot of work has been done during that leg, and still a lot of work to come with the analysis of all that data. Each of the scientific studies of the expedition is dedicated to a woman involved in marine sciences, and in this case it was dedicated to Tajir Bjornberg. Can you tell us a little bit about her and her contribution to the field of marine sciences?
So Tagea is a great teacher in São Paulo University in Brazil, and a true inspiration for all of us scientists. So she dedicated her whole life to study different groups of marine animals present in zooplankton, but her intellectual production is largely devoted to copepods. So again, another coincidence, I was in the lab and also I work with copepods. The main important work of her career was the one she conducted in Curaçao in the early 60s. Where she was studying some development stage, and then she had a problem in the lab work, malfunctioning, and she lost the circulation of seawater, and she lost all the animals died. She saw an opportunity to actually restart again, cultivating the animals and actually starting to observe all the stages. So she did this pragmatically to study the life cycles of copepods. So she's very known to describe it for her drawings of naupli and copepodites, which are stages before they become adult animals. So the value of Bjornberg's description for study of tropical and subtropical marine copepods, it's invaluable. Her work gained international attention after one publication in 1972 of almost 200 pages. So wow. It's a very descriptive work. All her drawings and descriptions in naupli and copepodites was published in the Journal of Studies on the Fauna of Curaçao. And still today, this work is widely cited, became a reference of development and behavior of these animals. So we can consider her as a pioneer in Brazil, and also with international recognized in the synthesis of knowledge in life cycle, feeding, taxonomy of copepods. And she is basically in the most important work that we have to identify copepods in the South Atlantic Ocean.
Wow, so really important, and again very related to what we do in Atlantico. And finally, let's talk more about your personal experience on board of Tara. Can you share with us what it was like for you? So the life on board, the memories you've created and brought back from that region of the Atlantic, and of course the people that you shared that journey with.
Yeah, so for me, being a resident scientist on board was a great opportunity to learn intensively, because I've been doing some sampling mostly close to the coast and also short time sampling. So this is quite challenging, but at the same time you are out of your comfort zone, you can learn a lot. So I might say that was the best experience of my life so far in my career. The best memory I have from this leg, it was after sampling station in the afternoon. We were above the Seamounts, and we have the opportunity to swim in the area. So while we were chasing the Seamounts, because the boat keep moving and floating, drifting, and the Seamounts peak is difficult to find because you have those 20-30 meters just in one spot of the ocean. If you are drifting too much, you are away like 2,000-3,000 meters. So we chased the perfect spot. So after some time, some of us actually observed the top of the Seamount. We have the nav guy on board, so he said that sometimes when they are in the ship, so they are higher than the schooner, in the water, if the water is very clear and you can see deep, you actually can see the Seamount. So we were like hearing his story and trying to think, oh, when we get the opportunity to see every day walking early. And so we swim around, and some of us could observe the tip of the Seamount reaching about 30 meters below the surface. And we could actually see some fishes spotting the area, which is very expected, and it's incredible, this vision. We are just so far off the coast, like 600 kilometers, and well, this was a fun after such an exhaustive work, the fun part. But I want also to say, I must highlight that the success of this leg was also largely due to the exceptionally good atmosphere that was established immediately among the entire group. So the seamen and the scientists, with a remarkably open and friendly and collaborative attitude of everyone. So I'm also very happy and pleased to say that I'm going to get on board again and see the Atlantic now in the other side. So I'm going to join the Tara and the Atlanteco team for a second round in the very last African leg, which is going to be now in August and September, from Gambia to Senegal. So I can't wait to be there again and do a great job for this amazing project.
Yeah, it sounds amazing, and maybe we'll have the pleasure to hear again from you and your experience on that leg. Wishing you all the best on your next adventure on Tara. It has been an absolute pleasure to talk with you today, and thank you for sharing your insight and your knowledge of the region, and with a lot of enthusiasm and passion as well. That translated really well. So thank you, Erica, and hopefully see you soon on the podcast.
Okay, thank you for inviting me. This is a very nice opportunity for us to share science. I think the knowledge can only be true understood for us as science, but for us as a society, when we can share experience and this create an awareness and a conscious, and I think through these we can make policymaker and change things that are not going well for the environment.
We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the Atlantico project on our website on www.atlantico.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes. Atlantico is a project funded under Horizon 2020, a European Union research and innovation programme.